Are we going? Are
There
we
we
sharing?
go. I can see it. I can see the share.
All right. So, hmm, AI synthetic genomics, a potentially $100 trillion opportunity. So the goal I have for today is that to give you insight into what's coming, why I say it could be a $100 trillion opportunity, why people like Suleiman has said it could be a $100 million opportunity. My promise to you is at the end, you'll be understand why I say it's a $100 trillion opportunity. And some people are going to make a ton of money. I hope everyone listening will be one of those. And I hope you can understand why my clients will participate in it. Why my clients in their accounts are doing 6 times better than the average financial advisor. That's the whole thing. But to understand that, I want to begin with the birth of the pharmaceutical industry. It really began as a function of the Spanish flu, 1918. Spanish flu killed 50 million people. Think about what that means. We went through COVID. We lost 7 million people because of vaccines. They didn't have a vaccine. They didn't know how to deal with Spanish flu. And one of the people that was working on trying to find a way to deal with Spanish flu was a guy by the name of Arthur Fleming. Fleming was a Scotsman. They didn't have big laboratories back then. He was working in his house. He was continuing to work and continued to work for over 10 years, trying to figure out a way to respond to and find something that would stop the Spanish flu. The Spanish flu was worse than the Black Plague that killed a third of all Europeans. 50 million people had died, and usually people were dead within 48 hours after they had their first cough. And as he worked for 10 years trying to find something that would deal with the Spanish flu and put it away, he was adamant, he was intense, and he continued to work on it. And about one time, he decides to go on vacation. He leaves a window open and he's gone for a couple of weeks. When he comes back, in the petri dishes that he was working with, there's a green mold. The green mold, of course, was penicillin. But penicillin was very, very difficult to manufacture. It was very difficult to make more than in a few instances. But Fleming was consistent and he used it to cure a policeman and a dog of the Spanish flu. He had discovered something that really worked. That was the birth of medical drugs as we know them today, as they came into effect, the big pharmaceutical companies. But penicillin was tough. Penicillin, 1928, it took another 17 years or 15, 16 years for penicillin to be made in a um, to be manufactured in any volume. And it was World War II when there was a big push on because soldiers were being wounded. The government and the military were looking for a way in which to treat the soldiers. And penicillin was one of those. It would be Pfizer that developed the technique to produce the drug in quantity. Think of it. 1918, the Spanish flu, 1928 before they discovered penicillin, and 1940s before they make it in quantity. And the way they found it was that they tested against trial and error against a number of different substances. And that's the way that pharmaceutical industry grew up. The Abbott Labs, the Bristol Myers, the Novartis, all those companies grew out of the same kind of trial and error. They had people that would go out into the world and look for natural molecules, inorganic or organic molecules and they built a whole library of molecules. Abbott Laboratories had a laboratory or a library of 14 million different molecules. Everywhere they found something that was different, they would culture it, they would grow it, they would duplicate it, and make it into one of the molecules that they would put in the library. And then they found a disease candidate. They would run the disease candidate trial and error against 14 million different by the 1980s and 1990s, 70s. They were running it against 14 million different molecules to see if there was any interaction with the disease molecule. And if they got a hit, and they would get a number of hits, then they had to take it with researchers and see if they could actually stop the disease and find a substance, a molecule that worked against the substance. And the process was a process of the researchers coming up and formulating a drug candidate to fight the disease, testing it in animals to see if it worked in animals, and then taking it into human trials. And the way that worked is the human trials were set up actually after the problem of thalidomide. If you don't know what thalidomide is, that's something to look into. But they set up a procedure coming out of the thalidomide process of doing phase 1, phase 2, and phase 3 clinical trials. Phase 1 was a trial of a very few people with the drug to see if it was safe. It was all about looking at the safety of the drug. Phase 2, if the drug proved to be safe, was the trial where they would run it against a number of people, but different doses to figure out what the correct dose was. And once they figured out the correct dose, then they would run it in a phase 3 clinical trial. And in the phase 3 protocol, they would have a treatment group and a control group. The control group got a placebo, the treatment group got the drug itself, and you test it against that disease in a phase 3 trial. The phase 3 trial was usually large, and you wanted to find a statistical improvement with the new drug compared to the old drug. This whole process, from the time you started in phase 1, 2, and 3, the success rate was approximately 10%. 90% of the drugs that went through either failed in phase 1, phase 2, or phase 3. Either they failed and were not safe, or 2, the dosage didn't really matter, or phase 3, the drugs proved to be ineffective. 90% failed. 90%. That was the process. That's where the pharmaceutical industry grew up, because through that whole process, they were able to find any number of drugs that worked against diseases. But the cost of putting one of those drugs through that whole process, the average cost of developing a new drug cost about $600 billion. That was a significant $600 million. That was a significant investment into a drug. But something happened in 1953 that would change all of that. In 1953, Watson and Crick produced a picture of and announced that they had identified the DNA molecule. They took a strand and made available how the DNA in our body was. It's what makes you and I unique. In some ways, it seems so simple because it was just 4 protein molecules combined in pairs coupled into a long strand. A strand that, if it was the size of a sewing thread, would be 150 miles long. Those pairs of letters are repeated over 1.5 billion times. And if each protein was a letter in a book, the book could be 1.3 million pages. You know, we're going to talk more about that. But that's what happened in 1953. But just as I talked about the beginning of the program, if you didn't see the forward, 1840, I talked about what was going on in 1840 and the early 1900s. Long time. Even if you look at the period of time from the Spanish flu, 1918 to 1928, the discovery of penicillin, and from 1928 to the 1940s, when it was commercialized and built in quantity, the biotech revolution took a long time. Stanley Cohen and Boyer, 20 years after the helix, the DNA helix was discovered, found that they could combine two pieces of DNA to make recombinant DNA. They combined DNA from different sources into a single molecule and inserted it into a bacteria. They showed that genes could be moved from organisms. They launched the genetic engineering of the biotech industry, 1973. And 10 years later, 1983, Genentech made the first synthetic drug, insulin. It was approved by the FDA. That was then. I remember going to a biotech conference in 1987. There weren't many biotech companies at that time. I attended the first one. It was in San Diego. I only remember 4 of the presentations, and I think there were 8. Genentech presented human necrosis factor, which we now call human growth factor hormone. Centocor was working with monoclonal antibodies for inflammatory diseases. Cetus discovered beta interferon, and Amgen was seeking to increase red blood cells. The only presentation that I really understood in 1987 was that by Amgen. Amgen identified their drug based on the erythropoietin molecule. Erythropoietin molecule was manufactured by humans. It was one of the molecules in urine, and it stimulated red blood cell production. And to me, that made sense. That molecule became known as, or the development of that molecule base became known as Epogen. And Epogen was geared for the replacement of kidney dialysis. The cost benefit analysis, patients whose had kidney failure and had to go through dialysis, that was an average cost of $100,000 per year. Amgen was selling their Epogen for $10,000, and it cost Amgen $10,000 to make. And yes, they spent hundreds of millions of dollars to get it there. They had a PaineWebber. It was a conference that was sponsored by PaineWebber, and the head of PaineWebber's healthcare stated that biotechnology was interesting, really interesting, but it was not going to make big returns. They were only going to make small returns. He said that biotech drugs had to be injected, and the only big drugs were orally administered. At the time of that biotech conference in 1987, there was only one drug that had sold $1 billion a year, only one that was injectable. Every other major drug was administered orally in pills. And he said, so biotech, interesting, fun things going on, but they're going to be small. I did, in fact, understand Amgen and their production and their protocols. And I didn't understand how it worked, but I understood it had a huge potential. And it proved to be a correct assumption. Epogen reached $8 billion in sales in the mid-2000s. Putting to rest that comment that it was going to be a small potential. the early biotech companies identified existing molecules like erythropoietin, and they modified it. By doing so, they replaced the trial and error. They began to look at recombining molecules to make new molecules. But they also took existing molecules from the human body. If you look what happened in the early pharmaceutical, it took 100 years, 100 years from the discovery of penicillin to get to where we are today. And most of that time was spent by trial and error. But that all began to change in the 1990s. In the 1990s, there was a company called Iguron, and maybe there were others as well, that began to look at what we can do, not to take a human molecule or an organic molecule. I know that people like Genzyme took molecules from a rooster to manufacture their drug, but they began to look at designing molecules, beginning to look at how they could look at a disease target and look at something that would connect with the disease target to stop it. It was Eguron, a company that I got to know way back when, and they began to design man-made molecules, man-made, that opened the pathway for a number of new companies to design. For Eguron, their initial drug was for Crohn's disease. They're too complicated a disease. Actually, Eguron built a drug later after that became a drug, Varicept, which was a major player in HIV. But it was a process of a man-made molecule, a molecule that didn't exist before, but combining human molecules. But as that's happened, and as that has proven to be a source of the new drugs that are coming, we stand on the verge of something additional. Because in designing a man-made molecule, you come up and you look at the disease target or the disease pathway. How does the disease progress? And try to determine how you can stop the progression of that disease. How can you arrest what's going on? Sorry, I forgot to silence my phone. That was a process of trial and error. Trial and error once again. A lot more sophisticated than the trial and error of the big pharmaceutical, but it was still trial and error. What we're approaching now is a new time in which we're really working into AI-driven future of drug discovery. Now we have reached the point where we can begin to design algorithms which grow themselves. It's no longer just a human interest into coding. It's where the algorithm itself will code and add code. I have a grandson. I actually have a son-in-law and a grandson that work in the high-tech coding industry. And my grandson says, I talk to fairly regularly, he says that as he's designing a program or an algorithm, he sets aside and asks a bot, an algorithm to code to meet this criteria. That's what's happening right now. AI is applying and building their own codes. It's no longer a question of humans building a symmetrical coding system. It's letting programs reprogram and write their own code to look at huge, huge databases. That's what's happening today. And that's where we are. So let me take it a step further. Let me talk about a high-level view of DNA and talk about it. This is what DNA looks like. And as we get further into this, I think you're going to understand how drugs are developed and what the future is. This is what DNA looks like. This is the double helix, the one on the right. This is the double helix. This is DNA. But if you put it into a regular format, this is what it looks like. These nucleotides, each one of these are a base molecule, a sugar, and a phosphate. And the A's are adenine. It's a protein with a sugar molecule attached to it. The A is adenine, T is thymine, and A and T are always combined. Guanine is always connected to cytosine. And all these molecules are formed with a backbone so that they go around and around. And they're small. These pairs are just 23 angstroms. If you put 1,000,000 of them side by side, they'd be less than 1/8 of an inch. And the strand is strung together in a chain that's always twisted to the right, always to the right. And the chains are always containing the same pairs in the same order. And the strand encodes between 20,000 and 25,000. And you may think it's a genetic work. Consider, humans have 20,000 to 25,000 genes. Worms have 1,796. But you know what? Corn has 32,000. Wheat has 107,000. So how is it that corn and wheat can have more genes than us humans have? No, it's not something to lay awake, sleep, keep you awake at night. But when you think about what DNA really is, it's the simplicity of 4 acids, and yet it can activate or repress actions in certain cells at certain times. There's a nearly infinite variation of how it's put together. Some single genes can cause other genes or combinations of genes to do certain actions. And just think of birth. Differentiation of cells. Ears are made only then and then do no more. The same with the nose and the organs and the fingers. And how do the 4 proteins make that happen? We're not even sure how that happens. Well, some companies going to figure out and so manipulate it and so manipulate the organs that they begin to replace themselves. Think about this. Heart disease kills 680,000 Americans every year. And I guess we're not too far away into the future when there'll be one or more companies that design and bioengineer a heart repair kit that'll replace a part of the heart that becomes defective, because the hearts do become defective. Our organs become defective. We're not that far away from seeing a company design a DNA that they can inject and inject the cell with DNA, a stem cell type of cell that's going to correct the problems with the heart. Think about it. Today, we're at a point where a lot of the genetic work that's done with the rational design of molecules, they're dealing with smaller and smaller populations. Just saw a drug approved for a population of 10,000 people. Heart disease, 680,000 per year. Think if somebody can cut that in half and what that does. But DNA is not just that. It's dynamic. It shifts things around. Cells immune to systems secrete antibodies that are designed to attach to invading pathogens. But pathogens are completely changing. We saw that. We saw that with COVID and the things that are going on. They change, they mutate. And you think of the word game. Maybe you've played a puzzle where you have to change the word hard to soft by changing one letter at a time. So hard, you change the A to an E to make heard. Then in herd, you change the R to an L to make held. Then the H is changed to an M to make meld, and so on and so on. If you've ever played that game, you know what it is to do. And that's what our genes can do. So there will be a company that will reverse that process. And instead of going from hard to meld, will take us from meld back to hard. And we saw that, a very glimpse of that in the RNA COVID vaccine. Think about what happens with diabetes. Diabetes happens when the beta cells fail and T cells destroy them. Imagine the company that can undo that mutation in the beta cells. Imagine the company that can undo the mutations in HIV. Those are things that are coming. Those are the things that we will see in the not too distant future. It's coming. It's only a matter of time. It's intriguing. I mean, if you look at chromosome 11, there's 155 genes that encode how we smell. It gives us the smells of spearmint, lemon, jasmine, vanilla, and pepper. And our genes regulate only one part of the 155 to transmit to the brain, so that each set for each smell is transmitted to the brain because we have genes that control that. You combine all those different smells and you end up with thousands of smells. That's what our brains do. Think about it. The potential that brings about by combining those genes, it's a difficult process. That's 100 understanding 155, what 155 genes do. And how do we really identify that? I heard a comment that the way that they identify what genes do is similar. An analogy can be made that writing a job description for a gene is like writing a job description for a factory that employs 45,000 employees. And you have to write the job description by looking at what happens in the production of that factory when one person is missing. You have to write that person's job description on what happens. That's how we determine what genes do and how they work together. That is what IA will enable us to do. AI has huge potential to do that, something that's way beyond human capacity. That's what AI is here. And for all of that, for all the genes that are in our DNA, they make up less than 2%. There's long stretches of DNA between genes. What do they do? We don't understand what they do. Is it a function? Is it just junk? You can imagine the Pacific Ocean with a few islands. That's what the genes and the DNA strand looks like. An ocean with a few islands. So the long-stranded DNA with a very few genes. Some from ancient viruses. And a lot of our genome is not even human. Apes have 96% of the same genes we have. We don't understand all that much right now, but we're going to learn a whole lot. We're going to comprehend how multiple genes spread across the genome coordinate. We know that DNA is used to build RNA, which is used to build a protein. How does that happen? How and why? We don't know. We don't even know the location of some of the genes. And AI is simply going to move us forward in that direction. When you think 4 little amino acids, the focus and the ability of AI to begin to identify what goes on in DNA is going to end up bringing us to a point where we have understanding of what's going on, it's going to bring us to a point where we develop new drugs, and it's going to develop those things which can repair hearts, that can repair organs, that can rebuild organs, can rebuild skin. Uh, all of that is the future. That's where we're coming. And it's not way off in the future. It's not that far away. Now, think about it. 680,000 people die from heart disease every year. 680,000. What happens when that's cut in half or cut by 90%? That's a huge potential company that can do that. Is that company formed yet? Not that I know of. The first company that is actually using AI to source and potentially source drugs is now formed and beginning that process. That's the first company, and there's going to be a ton of companies that follow it. Will the real focus of analysts and gurus really come to the same conclusion that the head of healthcare did at Payne Weber in 1987? That it's really interesting, but it's going to be a small approach. Or will they really say, this is going to be a significant development? I hope you understand. We aren't there yet. But if you look at where we're coming and what's happening, the potential is huge. My clients will be there. Will you? Will you? Anyway, with that, let me open it for questions and discussion. What are your thoughts?
There we go. Hey, Mike, that was a really insightful presentation. Thank you. Thanks for that. It's Chris Prouty. I'm back and I'm here to handle any questions that you might have. So we've got two different ways that you can ask questions. There's a Q&A function. You'll find the Q&A button at the bottom of the Zoom. And if you click on that and you type in it, that will be private. Only Mike and I will see that. If you do want to chat with us in the chat, the webinar chat, you can do that as well. But if you want that to be private, make sure hosts and panelists are is the one that's selected. Otherwise, everybody that's here will see it. Mike, a question that's come in is, are we talking about pharma companies who are implementing AI or AI companies who are serving pharma?
My guess is that big pharmaceutical companies will miss the whole thing. We're seeing even now with what's happened in bioengineering and biotechnology, that we're seeing some of the big pharmaceutical companies that are gasping for air.
Mhm.
They have not kept up with what's going on. I would imagine that we're going to see companies, it's going to be the synthetic biology companies, synthetic genomic companies
Mhm.
that combine with AI companies or AI use. There'll be new companies that come, just as Amgen and Genentech and all the bio companies that grew up at that period of time from the 1980s and 90s and 2000s, just as we saw those companies come, the big pharmaceutical companies could very well have entered those markets and had the ability to do it.
Mhm. Mhm.
But
Mike, next question is about timeline. Is this a 1 year play, a 2 year play, a 10 year play?
it's hard to know for sure.
Mm-hmm.
We're seeing the first company formed. I would imagine in the next couple of years, we'll see dozens of companies that are formed. When that, when will we see the first drug developed from those companies? I wouldn't be surprised at all if it's in the next 5 years. And the IPO for such a company could be in the next 5 years, maybe a shorter time. Things are
Thanks, Mike.
things are
Mike,
moving really fast.
Next, let's come in. Thank you for this information session, thank you for this informative session. My question is, can AI help determine the best therapy for chronic disease prevalent in different races?
Oh yeah,
Ah,
that that I didn't cover at all. But not only are they looking at drug discovery, I looked at just drug discovery, but AI is also going to be able to analyze what you do in your body. What
mhm.
they're going to be able to take samples of your body and tell you what potential diseases are there, what you can do. Um, it's AI is going to be able to predict how treatments, what treatments you should have,
Hmm.
what preventive medicine or what preventive care you should have. Um, I liken it to an analogy. There was a UK company that came out with a toilet, and I think that Japan is actually manufacturing it, where that toilet takes what you deposit in the toilet, develops what's missing in your diet,
Mm.
sends an order to the grocery store, and the groceries get delivered for what you should be eating and consuming. That's that's a simplistic analogy, but think of what it means for you to wear a watch or to wear a device that will monitor your physical condition and your body condition, alert you and alert your doctors and medical care people to say, this is something that needs to be intervened. There needs to be an intervention, or this person needs to have this prescription of medication, or this person is in risk. They're showing a certain increasing risk of what's going on. We get a sample of that. Get a sample. I'm a male. So I have, uh, um, I'm at the point where I forget what I'm saying.
Well, Mike,
Okay,
go ahead.
prostate cancer. 90 or 95% of men over the age of 55 will end up with prostate cancer. I had prostate cancer, but they monitor markers like PSA, and they could say your PSA level is increasing, and so we need to intervene and do a biopsy. Um, think about it, when that is for every disease we know, they can do that. And when you have a monitor that monitors your body 12 or 14 or 24 hours a day. And then, yes, it was a long answer to a simple question.
Yeah. Mike, I can add a little bit to that. Those toilets that you were talking about that do urinalysis, there's a city near me in Central Florida called Lake Nona. And in Lake Nona, there's an entire community of homes that they built with those toilets in, and they're all connected to the hospitals so that residents can have their urine analyzed in real time, which is pretty interesting.
Yeah.
Uh, in the webinar chat, first of all, someone had asked for Mike's email address, so I threw in a link. You can email Mike, and then I put his email address there. I know for a fact that Mike checks, reads, and responds to every single email that he gets. So that is there available for you. And then someone else asked if they could get access to this presentation. This presentation and this recording and all of our Thursday presentations can be found in our YouTube channel, which is About Money Radio. I put a link there for you for your convenience. And you can go there and you'll see this episode within a day or so once we get it authored down and uploaded. But you can see previous episodes there as well. And if you find value in that channel, would love it if you gave us a subscribe. And also, you could share that channel with somebody who might find value in it as well. Mike, let me check and see what we've got. Mike, somebody's asking if there are any other industries outside of pharma where you're seeing this much action from AI.
Oh, you're seeing it in a number of industries. And You're seeing it in robotics. Uh, Going to talk about it next week as well when we talk about it in manufacturing. Um, you're seeing it in software companies and data analysis. AI is one of those new technologies. Uh, I mean, something, if you look back in the 1970s when we began the first 60s and 70s, when we first began to apply computer technology to production, manufacturing, and to the service industry, it
Hmm.
was rudimentary and it was took a while for all that to happen. Next week, we're going to talk about it in terms of manufacturing, and I'm getting ahead of myself.
Hmm.
But yes, AI is being produced not just in this area, but it's being produced in robotics, in manufacturing, in many of the, it will probably be used in sourcing food sources. So,
Hmm. Hmm.
yeah,
Mike, Mike, somebody asks, is this better suited for growth portfolios or can retirees play here too?
it is a growth area,
Hmm.
um, but retirees should think about growth as well. I just have the statistics, by the way. Um, Let me share. Uh, if you're 82 years old, and I am 82 years old, the average life expectancy at this point is another 18 and a half years.
Mm.
If you're 75, the average life expectancy is another 24 years.
Mm.
If you're 90, it's 12 years. What that's saying is if you're 82 like I am, my life expectancy is another 20 years. The old ideas of a stock portfolio that's equal to your, or the stock bonds should be equal to your age and stocks equal to your age minus 100 was really workable when in my father's day, because when my father was my age, had been dead for 21 years.
Mm.
When my father was coming to retirement, he was 61 years old. When he was coming and considering retirement, he wanted to retire at the age of 62. One of the options offered by Pacific Gas and Electric, and he worked for Pacific Gas and Electric, one of the options was a 5-year annuity
Mm.
because most people that retired at 62 or 65 didn't live beyond 5 years.
Wow.
Today, my life expectancy at 82 is another 18 years, over 18 years.
Mike, that's a lot of Thursday workshops we'll be doing together. Mike, I have a question for you. Can you give us the title of next week's presentation?
Sure, let me go there. What's happening with manufacturing? What's happening with jobs, automation, onshoring, wages, and how to profit from the changes?
Wow, that sounds like it's going to be a pretty full presentation. I'm looking forward to that. And everybody who's here, keep an eye on your email for an invitation to that workshop. And you'll see that coming through Monday and Wednesday of next week. And so you can block off this time on your calendar and we'll see you then. Mike, that wraps up all the questions that I have for today. I want to thank everybody for being here. I'm really grateful to see everyone here and what a great presentation it was. Mike, I'll hand it over to you for the final word.
Thanks for being here. Um, Next week, I think, is going to be a very interesting presentation. And I will also offer, if you would like to talk about your portfolio and how you can benefit from being part of us, I think you get some indication of what we do and why we outperform most financial advisors. And if you're 82 and have 18 years of life left, it doesn't take a genius to figure out what prices will be in 18 years and why some growth is needed in your portfolio. And with that, have a very good week. Pitchers and catchers report in 2 weeks, and I'm very excited about that. Have a great great week. I'll be back next week.
Thank you very much for being here.